Hello! In today's lesson we're going to have a look at the tensile strength and the different uses of plants. So we need to start with the different uses of plant fibres. We're going to explain why some of them are going to have a higher tensile strength than other ones, and in the end we need to plan a practical use to investigate the tensile strength of plant fibres. So let's kick off. Now you might recognize some of these textiles already or different materials we're looking at, and these are very common plants that we use in order to get their plant fibers from. Usually it's from the stems, sometimes it can come from the leaves, but we have flax plants, we have cocoa coir from coconuts, we have hemp, we have jute, and we have manila, which is from banana leaf fibers. So, what we need to think about is, what do people use these for in the world? Take a moment, pause, open a search engine and see if you can find a few common uses of these. So starting off with flax and cotton. Flax is different because we're using the stem, whereas in cotton we're using the fluffy hairs on the seeds. And both of these are used in clothing, flax to make linen and cotton to make cotton. Next we look at hemp, and hemp we can be using for ropes and carpet backing usually. Coir is used in floor coverings and ropes. Jute is from the stem, and that is used in hessian sacks and carpets. Manila from bananas are used mostly in ropes again, so marine cables, nets and matting. And then pulp from softwood trunks is used in paper and cardboard. So there's a whole range of different reasons why we might use plant matter, but in a summary, we use them for clothing, we use them for ropes and nets, paper or cardboard, or to put on the floor. So clearly there's something in plants, in their stems and leaves, that helps them be so strong and supple. So think about what we know already from our previous lessons. In the stem or leaves of a plant, what's going to be running through them? What materials are there that can increase the strength of a plant? Think of where these materials can be found. What common structures might you find these materials in? Take a moment, pause the video, and we'll go through in a minute. So, when you're having a think, hopefully you came up with these two materials. We have sclerenchyma tubes and we have xylem tubes, and in the cell walls of each, you can see them there stained in pink, is lignin. Now lignin is a component that you find combined with cellulose in the cellulose cell wall, and they are thicker in those two vessels. There is more lignin linked with the cellulose, and this allows it to be stronger, flexible and more waterproof. So if we look over onto that picture on the right hand side, you notice that there's a vascular bundle with different tissue layers. So here's the whole vascular bundle, and we can find the xylem up here, the phloem in the middle, and sclerenchyma on the bottom. So this is where we would be looking for the stems and leaves in particular, as we'll find a lot of vascular bundles together. Now when we think about what sort of properties we'd be combining and comparing in plant fibres, you've got three different plants shown. You've got a fibre section from each plant, and we could just look at this picture for a minute and pick out different things we could look for. So there's a number of different properties I might compare. You might decide to look at each individual cell wall and be making observations of which is thicker and which is thinner. You could compare the diameter of the lumen in the different sclerenchyma or xylem vessels. Or you could just do a simple count of the number of vessels that you find in an equally sized piece of plant tissue. But really, the experiment that we want to do and get familiar with is investigating tensile strength. So tensile strength and tension is linked to pulling. So if you look at this picture, figure number 2 here, you notice that you've got a stick or a twig beam, and it's clamped at 2 points so that it can't move, and tension is applied by suspending a weight off the center, and which applies force. And what you would expect to see is the more stress that you cause, or the more the more mass that you add, you might break that twig at a certain point. So let's look at how we would do this experiment for biology. Well, first off, we need to get the plant fibres out of the tissue. To get the plant fibres out of the tissue, you can use a process called retting. You can do this in fields. You could harvest a little sample yourself while you go on an exercise walk and treat them but we start with adding water or adding a solution. So in the industrial method, you don't need to wait quite so long. You can just treat your plant stems with an enzyme solution to digest all the green fleshy stuff around the xylem and sclerenchyma. Or you could just soak them in water for a long time where you've got lots of bacteria and fungi, and they would do the work for you, and they digest all those fleshy green bits and leave you with that last picture that looks like different strands. So only the harder lignified plant fibres are left that you would find in your vascular bundle, xylem and sclerenchyma. So once you've got your plant fibres, you can start to plan a practical. And if we were in college, we would do this together. So instead, let's have a look at the method and the principles that guide you planning it. We're going to look at comparing 3 different types of plant fibres from nettle, New Zealand flax and celery. And we're going to investigate the mass or force required to break those fibres, and that will give us a measure of the tensile strength. So we start with a hypothesis, then we look at the apparatus that you'll need in your equipment. You'll look for a method that will produce valid results. We'll look for the independent and dependent variable, identifying controls as we go through, making replicates and explaining why we need them, and then running through what observations and how we're going to make sure that we are valid, accurate and precise. And finally, before you could carry it out, you would absolutely need a risk assessment that picks up on any safety issues and risks that you might encounter. So after this, with some dummy data, later we'll look at graphs and how you'd present it, make conclusions and draw an evaluation. But today is more about planning. So let's start with our hypothesis. A hypothesis is a statement or an assumption that your experiment is going to look to test. Usually we have 2 in science. We have a null hypothesis that predicts no relationship between the independent and dependent variables. So for example, you could say there will be no difference between the plant tensile strength and the materials used. Or there will be no significant difference between the length of plant fibres used and its tensile strength. Or you could look at the alternate hypothesis, which is assuming there is a relationship between the 2. So you could say there will be a significant difference between, or there would be an increase between, or a decrease between your 2 factors. Next, we look at your apparatus and think of a method. So let's plan some of it together, but ultimately I would like you to write it down as you go along. So we're starting off with our apparatus laid out the way shown, where you've got your 2 clamp stands, and each clamp has got a bit of plant fibre suspended in between, held nice and taut. Underneath it you have a soft surface to absorb the impacts of any masses that are going to fall, because using a mass hanger you can add increasing weight to this, and then you record the measurement that the plant fibre snaps at. So you need to think about what increments you're going to go up in. Are you going to add 50 grams at a time, 100 grams, 500 grams? When you add your masses, are you going to need a timer? Will you need to count how long you are adding the masses for each time? Or are you going to just keep adding masses suspended until you see the break? So in your method, you need to think about what you're going to keep the same and how you're going to set this up. What order are you going to do these things in? Next, we are looking at the independent and the dependent variable. So we know the independent variable is the factor being manipulated or changed. This is what you're doing, the conditions that you're setting up. And the dependent variable is what you're measuring as a result of your experiment. So if I'm choosing 3 plant fibres, nettle, flax and celery, is that the independent or the dependent? Your control variable is a factor that might influence the results, so you either keep it the same or monitor it if you cannot change it. So these are things other than your dependent variable that are going to be affecting your results. So think about the different conditions of the plant tissue or of the surroundings that might interfere with the structure of your plants. Next up, we are talking about the control experiment. So this is where you're not measuring your dependent variable, you're not doing things to your practical. Instead, you have a control which can be used to compare your results to show the effect of other variables. So let's think of some things that we might have to control. If you look, we've got two different plants that we've extracted plant fibres from, and the plant fibres are of varying sizes, even within the same type of plant tissue. So would it be valid to compare these results if we know they all have different diameters? What might be better? And then we need to think about something else. What if some of the plant fibres in one plant were wet and the others were dry? How would that affect things? So let's take a moment to try and think of a few reasons this happens. Let's look at temperature, the length of the fibre used, the age of the fibre, and humidity as all things that can affect the tensile strength. So pause for a moment, and I'd like you to look for an explanation of how would the cell wall structure be linked to temperature, or the length of the fibre, or the age of the fibre? How might humidity affect how much weight this plant could withstand? So first off, temperature. Now you might know from the experiments we've done earlier in biology that temperature has an effect of denaturing proteins or affecting the bonding and the structure of different molecules. So if you increase the temperature or decrease the temperature extremely, you might be able to affect the bonding between the polysaccharides in the cell wall. So that's your cellulose. It might be talking about the substances you find in the middle lamella. It might be talking about the hemicellulose that you find running through different cell walls. So between pectin cellulose and hemicellulose, you might mention something to do with those. Alternately, you might be thinking about your lignin that you find in your sclerenchyma. So all of those polysaccharides might have bonds that are affected, weakening the structure of the xylem and the sclerenchyma. Next, thinking about the length of the fibre used. So if you use 10 centimeters fibre for one test and 5 centimeters for the next, varying the length might affect the amount of tension that plant can withstand. So one plant fibre might be more able to bend and cope with the tension than another. So this might have an impact. Next, the age. Older fibres might be more brittle and less flexible, less able to bend, meaning that their tensile strength again is being varied and you're not measuring the effect of the two different species. And lastly, humidity. So different plant fibres from different plants might be affected differently by levels of humidity, or even if you have one type of plant and some fibres are wet and some fibres are dry, this could cause inconsistency in your results. So this is something you would need to control. The next factor is thinking about repeats and observations, and thinking about why you would do those repeats. So I want you to think about why you wouldn't just do one trial for each condition and explain the significance of it. What does it allow you to do? For observations, let's think of a results table that you might do. I've given you a template here if you'd like to use it, otherwise have a go at your own. So now you've tried to write your own observation table, we can have a look at one that I've modelled for you. So we're going to put one variable down this side, and in an earlier question you need to identify whether that's the independent or dependent, and likewise we put another down the other side. And we have our columns for our repeats, and we have a mean mass category at the end, so think about how you could work that into your answer. Next we have quality. You need to be able to justify why the accuracy, validity and precision is ensured in your own method. So let's look at the definitions for each. If you're talking about accuracy, we're talking about recording values close to the true value or the standard. So if there is a known mass of something that you need to get, how are we going to accurately measure that? For example, you can calibrate equipment, you can choose equipment that's more able to measure true values, you might repeat measurements and so on, using different pieces of equipment in order to make sure that your results are accurate. An example of results not being accurate is if you're in one room using one scale and you take a measurement of 9.8 kilograms and then you use another scale to measure exactly the same thing and you get 11.4. So think of what you would do to make sure that your readings are accurate. Next we talk about validity. And a validity describes how well a study measures what it's actually supposed to, what it intends to. And one of the best ways of ensuring validity is to control other factors so that you're restricting the conditions of your experiment to such that only the independent variable has an effect on the dependent variable. Lastly, we have precision. And precision is about recording reproducible results that are closer to one another. So if you do repeats, and each repeat gives you similar readings, then your data is more precise than if you get very different readings for the same type of plant fiber, for example. So in your method, how are you going to ensure that your results are accurate, valid, and precise? And the last thing that we need to consider is a risk assessment. We need to identify things that could pose a hazard to you and what method you use to reduce that risk. So identify any substances, any equipment, or anything that could happen that has a negative consequence for a person's health and safety, and I pop that as a hazard. And then you need to describe how you're going to reduce the risk for it. When you're done, if you're in my class, you'll have a table that you can download. You can pop all your answers in and upload that to Moodle for me. But now let's have a look at some practice questions that simulate the sorts of things that you could be asked about this practical. So we'll start with this nice graph. Here you have on the y-axis the mean tensile strength in megapascals, and you have the fiber diameter in millimeters running along the other axis, the x-axis. Using the information in the graph, you have to describe the relationship between the diameter and the mean tensile strength of the fibers. So, this is a 3-mark question. Let's identify any trends that you see, if there's any significant gaps between places, and give it a go. So overall, looking at our data, we notice one thing very immediately. Starting from this position here, we can go down, and we notice that there is a significant drop as you go across. So, the way to explain this would be to mention exactly what happens using those axis labels. The larger the fiber diameter, the less the tensile strength. This could be also described as a negative correlation, as one factor increases, the other factor decreases. We could identify other key points. So if you notice between this column 0.05 and the 0.09 that there is this huge drop between the two. This is significant. This is a relationship between those two factors, so we can describe it. So you could say there's the greatest decrease between these two columns. Following a similar line of thought, we could look at the rest of the data, which seems fairly consistent. So you could say that there's little change between 0.13, so this column here, all the way up to 0.33, which is this column here. So I could just draw an arrow there to show that they're all fairly consistent. If you've not said anything else, that's fine, you'd get your 3 marks, but you could also mention that there's this very slight increase between this column and this column here, fibers of those diameters, and you could also manipulate some data, so you could calculate a percentage, or you could give a number to show how many units they are different between that 900 and this 500, let's say. So, looking across to the next question, you need to suggest which variables should be controlled when you're investigating the strength of fibers of different diameters. So, if we're changing the diameters as part of our investigation, we can't have that as one of the controls. So we need 3 different things. Pause and give it a go. Okay, so the other factors we could have talked about are length or mass or an equivalent point. You can mention the age of the fiber, the source of the fiber, so do they come from the same plant, do they come from different plants, the temperature and the humidity of your surroundings. Give yourself 3 marks. Next, let's have a look at this picture of sclerenchyma tissue. So the photograph below shows a group of sclerenchyma fibers labelled S. There they are. You need to use information in the photograph with your own knowledge to suggest how the structure of sclerenchyma makes them useful for making rope. So what do you know about the properties of sclerenchyma and its structure? What makes it up? And as a function, making it useful for making rope. think about those cell walls, think about what's inside, what makes their structure. Give it a go for 2 marks. So the first thing that should have immediately jumped into your mind is that we have lignin and we have thick cell walls to give it added strength. Because lignin's present, we also know lignin makes them waterproof, and if it's waterproof then it is more flexible, it's got the strong cell walls, and it won't be able to break easily. But also looking at the photograph, you notice that sclerenchyma are hollow, so that makes them lighter. So any of these, any 2 points out of this, is what you need to explain why it's useful as rope. You could also have a few extra points really. So if you didn't say it has thick walls, instead if you said it has tough walls or the plant cell walls are tough, you could also have a mark there. Now we have a lovely quick multiple choice question. Textiles are often made from plant fibres. The use of plant fibres is more sustainable than the use of synthetic fibres made from fossil fuels. The tensile strength of plant fibres from different species can be measured. Which of the following are variables that would need to be controlled when testing the tensile strength? So look at these different pairs. We have humidity and light intensity and temperature, temperature and humidity, and temperature and pH. You need to identify which will have an effect, and you do need to control them, and which do not have an effect. Give it a go. So let's look through our answers for this question. If I get my highlighter out, we can pick up on the ones that we feel familiar with. So we've mentioned humidity before, but we have not mentioned light intensity. We've mentioned temperature having a potential effect, but not light intensity again. We've mentioned temperature and the humidity, and we've also mentioned temperature here too. So even though light intensity and pH might be factors that you think is important for a plant, in this question our answer is C. Only temperature and humidity are going to have an effect on tensile strength. I hope that's been useful for you. As always, email me any questions, my class, if you need a hand, and we look forward to seeing you next lesson. Bye bye for now.